Welcome to another report commentary with the CEO of Herantis Pharma, Craig Cook. Welcome back, Craig. How are you?
Thank you, Kaiser. Fine, and you?
I'm great, thank you. So let's start off with the announcement that you made yesterday on the 2nd of March, that the results from the phase 2 study with Lymfactin were inclusive. What happens now?
So Lymfactin is our gene therapy that induces expression of something called VEGF-C, which is vascular endothelial growth factor C, that is responsible for driving the growth of lymphatic vessels. And in breast cancer-related lymphedema, which was the indication or is the indication for Lymfactin, patients that have breast cancer, when they come in for treatment and they have the mastectomy, at the same time, they often have the lymphatics removed from their armpit as well at the same time to prevent spread of the cancer. But removal of the lymph nodes and the lymphatics from the armpit means that there's nowhere for the lymph fluid and other fluid in the arm to drain, because the function of those lymphatics is to drain fluid from the arm back into the body, just like the veins in the venous system in the arm take blood back into the body. With lymphatics, they're taking fluid back into the body. So when those lymphatics are disrupted, then there's nowhere for that fluid to go. The arm becomes heavy, it's painful, it's swollen, it's debilitating for these patients. And to correct this or to treat this, in severe cases, patients require surgery to repair the lymphatic system. And the primary purpose of the study that Herantis has been running was to determine whether there was an additional benefit of adding Lymfactin to the surgery that these patients would normally have. So we had two groups in the study. We had a group who just had surgery, and we had a group who had surgery together with Lymfactin. And there were two main outcome measures as well in the study. One was volume reduction, to see how the volume of the arm reduced as the lymphatics were replenished. And the second one was quality of life improvements for these patients, because obviously this is a big impact on their quality of life. And in the study, what we observed is that while both treatment groups overall experienced clear clinical benefits, it wasn't possible for us to draw any conclusion specifically about the incremental benefit related to Lymfactin. And there were two main reasons for this, it appears. First of all, remember I said there were two groups, one with surgery and Lymfactin and one just with surgery. Now, both of these groups at baseline from the start of the study were very different. And in a study such as this, it's important to have those two groups comparable at baseline so that you can compare the treatments. But in the study, it's become clear that the patients, the two groups of the patients were very, very different. The second factor is the outcome measures that were used in the study, which was looking at reduction in arm volume and improvement in quality of life. And the measurement of these that we observed in the study were very inconsistent and unreliable. So we had two issues with the study. One is the comparability of the baseline of these two groups at baseline, and the other one is unreliable endpoints or unreliable measurements of endpoints in terms of arm volume and quality of life. Both of these were somewhat unexpected and surprising, and we're not fully sure of how it came about. And even the statisticians and the investigators on the study were similarly surprised with these findings. But now moving forward, we are going to do a deeper analysis of the data, and there's a piece of work planned for the next 4 to 8 weeks, where we will take a deep look at both the clinical trial, the data, as well as the Lymfactin program. And once we are through that piece of work, then we will update the market on how we intend to move forward. So sorry, that was a long answer to your question, but I think it's important to capture the full context.
I understand. Is it likely now that when you know these issues, that you will redo the study?
Well, that's going to be the focus of this piece of work that we're going to look once we've gone into detail in the data, completed the analysis on both the study, the data, and the program. We will then be in a position to make a decision on how best to move forward.
So let's move on to your other candidate, CDNF for Parkinson. You have a new strategy here. Can you tell us more about this, please?
Yeah, so you may recall that the original phase 1 study, phase 1/2 study with CDNF used a very invasive intracranial surgical device for administration. And basically patients needed to have a hole drilled in their head, in their skull, and then a complex system was inserted into the deep areas of their brain. And concerns with this route was that it limited the use of CDNF to very late stage patients, number one. Patients didn't like it. It's not very patient friendly. Regulators had their concerns, and they forced us to focus on late stage patients. Potential partners, pharmaceutical companies, this was a concern for them as well. So in parallel to that, we evaluated other routes of administration that wouldn't require surgery. And what the new strategy is built on is that moving forward, we have 3 key pillars of success, or the program. Number 1 is that we are going to back CDNF as a standalone product. So we're going to move away from the need for surgery, and we're going to focus on 2 alternative administration routes. One is intranasal or nose to brain, so just administering it via nasal spray. And number 2 is via skin, via a skin injection, a subcutaneous injection. Both of these are already ongoing programs in Herantis for a couple of years. We have data to support that, and we know that we can administer CDNF via these routes. So that's the first key pillar moving forward of the strategy. The second key pillar is that we will be accelerating development. And the reason for that is because we don't have this complicated invasive surgical device in the picture anymore. So that means we don't need expensive studies, we don't need such long studies, we don't need such difficult studies. Regulators won't require us to jump through lots of different hurdles and obstacles. So overall, we're going to accelerate the development by just focusing on CDNF as a standalone product without having this complicated or invasive surgical device in the equation. And the third key pillar moving forward is that, as I mentioned earlier, we're going to expand the target population to include earlier stage patients. And this is important because the mechanism of action of CDNF acts when neurons are still viable. They may be declining, they may be deteriorating, they may be dysfunctional, but that's when CDNF comes in and turns around the pathology of the neurons. Once the neurons are dead, like in late stage patients, then no therapy, not even CDNF, can change things. So we've got to get in there while the neurons are still viable. They may be damaged, but when they're viable, we can turn things around. So it was essential for CDNF for us to focus on earlier patients and expand the target population to earlier patients.
I understand. Interesting. You also have a project in a preclinical stage, xCDNF. How are they different from each other, and how has this project developed during the last quarter?
So CDNF and xCDNF are quite different molecules. CDNF is a natural biological protein that occurs naturally in the body and whose natural role is to protect neurons. xCDNF is an engineered synthetic model where we've taken the smallest, most potent fragments of CDNF, of the parent molecule CDNF, and engineered that into a new chemical entity called xCDNF. And CDNF is a clinical stage asset, whereas xCDNF is still in preclinical. And although they're distinct standalone programs, there is synergies between the two programs, because both programs effectively are pursuing the key objectives to cross the blood-brain barrier and effectively treat the pathology of neurodegenerative diseases in the brain on the other side of the blood-brain barrier. So xCDNF is a very active and intense program, very energizing, and our latest data shows impressive effect on neuronal survival, on alpha-synuclein reduction. Alpha-synuclein is a bad protein that is responsible for a lot of the disease that we see in Parkinson's, or the pathology that we see in Parkinson's disease, Alzheimer's, and things like that. And we are also seeing biomarker findings with xCDNF, where there's a reduction in markers of disease as a result of xCDNF. And we'll communicate this in more detail later this year, but it's a very active, very intense program that's ongoing, and very, very much look forward to developing both of these programs with their respective merits.
Craig, during the last year, you raised 15 million euro. How far will this money take you?
The current runway extends into 2022. Yes, it was 15 million. There was a fundraise first half of last year. Then there was the fundraise in December to give us a total of 15 million, and that'll give us a runway into early 2022.
And before we wrap up this interview, what are your goals for this year?
Well, the last year or so has been a key focus, working on the fundamentals of our programs and portfolio. This year, we will continue with that optimization and shaping and preparing for the future. Related to this, we have rich news flow planned for the next 12 to 18 months, and our focus is on building value in the assets and portfolio, specifically for CDNF. In Parkinson's disease, we're going to complete the intranasal formulation, and we're going to enter preclinical later this year. For CDNF, with the subcutaneous administration, we already commencing preclinical programs with that formulation in stroke or reperfusion injury as the target indication. With xCDNF, we're going to complete the formulation and enter into further preclinical studies. And then, of course, with the Lymfactin, we're going to go through the data in detail and decide on the best way forward for the program. So a lot happening and rich potential news flow planned for the next 12 to 18 months.
Indeed. I want to wish you good luck moving forward, and we'll speak again in 6 months.
Thank you, Kaiser. Appreciate it.